just shared my screen so that as we go through the tutorial I can demonstrate things to you in case people are having any problems with running OpenCOR. So what I'll do is work through the parts of the tutorial that hopefully people have downloaded. I've got it up on my screen at the moment, and I'm not going to be able to get through all of this, but I'll get through hopefully enough that you can begin to use OpenCOR and access the Physiome Model Repository. And then the tutorial is reasonably self-explanatory for a variety of other more complex models, but so we'll start off with some very simple models just to get used to the features of OpenCOR. So on page 2 of the tutorial, there's a little bit of background to the VPH Physiome project that I thought I'd quickly go through just to give you the context for the what I'm going to talk about. So well over 10 years ago, probably 15 years ago now, the International Union of Physiological Sciences, IUPS, decided that it needed to pay more attention to computational physiology in terms of handling complex physiological problems and also being able to connect to molecular biology. So we began a project called the Physiome Project, which was intended to develop standards around the use of models, computational models, in being able to bridge molecular biology through to tissues and up to whole body scale physiological systems, organs and organ systems. And then, so that began in 1997, and then in around 2006, the European Commission, under Framework 6, commissioned a study led by Marco Visconti that produced a report called the STEP proposal, a strategy for a European physiome, STEP, and that led to the funding under Framework 7 of a whole range of projects based around computation for the health sciences. And the focus with the VPH was always both trying to link to clinical applications, but also to support and use the work that had been done under the Physiome project, and also to encourage industry links for the various uses of models. So that together now we call the VPH Physiome project, and a few years ago, at the end of the first phase of the 2007 Network of Excellence, we instituted the VPH Institute. We formed the VPH Institute to carry on the momentum associated with talking to Brussels and providing European but now global leadership in terms of Physiome VPH initiatives. And this also now involves the US through the IMAG. NIH began a interagency activity called IMAG, led by Grace Peng, which meets annually, and they, in fact, they're meeting in Washington around now, and they also are contributing to the overall goals of the VPH Physiome project. So that's the background, and one of the very early initiatives that we began that was fundamental to the Physiome project was the idea of developing standards that will facilitate multi-scale modeling, because it's use having people developing models if others can't use them and reproduce what they've done. So the development of initially CellML and then FieldML as a spatial standard. CellML is designed to handle ordinary differential equations and algebraic equations. FieldML is designed to handle spatial finite element type modeling. And there's also an initiative called SBML, started by the systems biology, molecular systems biology community. So at the level of lumped parameter models based on ordinary differential and algebraic equations, the two standards in common use now are CellML and SBML, and those two communities work closely together because they're very complementary. SBML is targeted mostly at biochemical reactions. CellML is targeted more at a broader range of biophysics and the link to particularly the link to multi-scale physics at the organ level. So if we go to the next page, one of the projects that we began a number of years ago was developing a software environment for creating, simulating, visualizing CellML models, CellML being the encoding standard. You want people to be able to fairly easily create new models and run them, and then be able to archive them into a repository that when models are published, they can be made available to the public. So we began the development of software and also a model repository called the Physiome Model Repository, and that has all the current CellML models, the 600 or so CellML models that have been coded up from the literature are on that repository. And I'll talk in a minute about how to access that repository. But just to get people going, I've suggested to everyone that they download OpenCOR. This is a software program written by Alan Garny, who lives in the south of France, and it's the primary tool that we use for solving CellML models. So if you have downloaded OpenCOR, what you should see is a blank, or at least a page with an icon displayed like this, and this should have, you should see two environments. One is a simulation environment and one is an editing environment. So we'll move between those two environments in a minute. And then also if you use the toggle, which on a PC is Control and Spacebar, or on a Mac is Command and Spacebar, you can toggle between being in an environment where you've set up your model and you want to run it, and an environment where you're accessing the databases of models either from the Physiome Model Repository or from the models that you've created yourself or you have on your local desktop. So if I just go back to the page here, I'm not going to do this, but you can move the windows around to choose whether you want to have them like on the left of that diagram or on the right. But for most screen shapes, it makes sense to have it as shown on Figure 1A. But you can dock these windows as you wish. The software is written with a plug-in architecture. It's C++ software with a plug-in architecture to keep it modular, and it uses Qt as the visualization environment, the GUI environment. So you can choose which particular modules you want to incorporate into your current version, but by default it makes to just leave them all on as they are when you download the software. So if we then go to the next section, page 4, and we'll set up a very simple model, which is a second order model represented as two first order models shown here, and I'll show you how to set those up in OpenCOR and then to run the model. But before I do that, maybe we should just double check there aren't any questions. Jerome is going to monitor any questions that come in, and then if we need to pause and deal with some of those questions, he'll let me know. So Jerome, any questions in so far?
So, okay, so apparently there are none in questions, no type questions, no hands.
Okay,
so
that's good.
maybe
All right,
we'll
we'll
go forward.
on. Yeah. So if you now go to, if you keep this page open on your computer with this part inside the box, is the part we're going to implement into OpenCOR to run the first model. And you'll see that this is it's a Van der Pol oscillator, the model. So the model, it's arbitrary what we call it, but I've called it Van der Pol model. and it has a component. Comp there stands for component, and the component that we're dealing with is the main component. So every model has to have a main component. And then we declare certain variables, time, and because CellML uses dimensions, all variables will have dimensions, whether they're dimensionless or not. So in this case, we're treating this as a dimensionless problem, so we have to say that it's dimensionless, and we give it an initial condition that time is initially 0. And we've got two other variables, x and y, so that second order equation was turned into two first order equations declaring y to be dx dt. And we give x an initial value of -2, y the velocity an initial value of 0, and there's one constant, mu, which will just give the value 1, the initial value 1. Then those are the two differential equations. So in OpenCOR, they are represented by typing in ODE bracket x comma t close bracket equals y, 1, and as shown for the second one. So let's do that so that everyone is up to that point. And the way you do it, if you go to your OpenCOR page and go to edit the editing environment, you should have click on CellML text if that's not highlighted. So this is the environment into which we're going to type these simple expressions to express the mathematics that we're dealing with. Then if you go to File, under New, open the file CellML 1.1 file. The distinction between CellML 1.0 and 1.1 is that 1.0 all models are flat, whereas in 1.1 we can create submodules and import them, which is a better way of handling a hierarchy of models for a complex model. But you can always, having created a hierarchy of models by importing, you can always flatten it and generate a 1.0 model. So what you should do now is simply type that code, and you can copy and paste if you want from here directly into the text, CellML text view. I've actually got a version of that here, so I'll bring that up. So that's what it should look like. You should see a code in the CellML text view under the editing menu item that shows some sort of model name, van der Pol, the main component, the declaration of the variable x, y, and the parameter mu, and then the two differential equations. And you know that you've got those equations typed correctly when you click on that, you can, it appears above the text. And so that second model appears, second equation appears there, dy dt equals mu times 1 minus x squared times y minus x. And note that every line here has a semicolon ending it. So, and if you save that, if you do Control S and save that, if it is able to be saved, it will. If there's a mistake in the code, it will give you a warning. So for example, if I was missing that semicolon and I try saving, I get a message saying that it can't save it. So maybe I'll pause there for just a couple of minutes to let everyone get the point. Typing those, copying and pasting those equations in. One thing to be aware of is that in this documentation here, anything, and inside OpenCOR, anything that's preceded by a double slash is a comment. So if you're copying and pasting from the PDF across to here, just take out that line involving the comment. I mean, you can have it in there, but it perhaps is a little bit confusing. Okay, so at this point, I'll carry on unless anyone raises a hand, but you should have those equations typed there, save that to some local directory. Once you've done that, click on the simulation view, and now we've got, you may have one window or more. You can vary the number of panels for displaying graphical output by using the plus and minus buttons. So for this model, I suggest you create 3 graphical output panels. Then the first thing we're going to do is to change the default time range. Instead of being 0 to 1000, we'll make that 0 to 100. And instead of making the point interval 1, we'll make that 0.1 for this particular model. The integrator being used here, if you solvers, you'll be able to see that the integrator by default being used is CVODE, which is a Lawrence Livermore very efficient ODE solver, open source solver that's incorporated into the OpenCOR platform, and it will automatically choose steps to achieve whatever error tolerance is specified here. You've specified, but those normally have sensible default values, so you can just leave them as is. So the point interval is mainly for plotting purposes here, just so that we get a nice smooth graph. So then the next thing to do is to click on, you notice that when you click on these panels, a blue bar appears next to the panel. So when that blue bar appears, that's the one we're dealing with. And we now want to allocate a variable to that panel. So if you go to the list of parameters listed here, you'll see X, and if you right-click, it gives you an option to plot against the variable of integration, which is time. We go to the next one, again right-click, and this time on Y, plot against variable of integration Y, and on the last one, we'll plot Y against X. So this is now a state space display. So then instead of choosing plot against variable of integration, choose plot against, and then from the list of main component variables, you can choose X. The symbols shown here have a meaning. So something that's red with a hole in it, the hole means you can edit that variable or parameter. The red means it's a parameter. The blue ones are the variables, and you can see that you can edit the initial value of X because it's got a hole in the middle. One that's a full here, a derivative, you can't edit the initial value of that because that variable is a Y. Okay, so you should now have, and if you list under graphs, you can see, sorry, I'm going to remove one because I had that there for a previous model. You should be able to see, first of all, that you're plotting X versus time, and then Y versus time on the next one, and Y versus X on the bottom one. So then if you just click the little arrowhead button, Run Simulation, the simulation runs. It's very fast. CVODE is a very efficient time integrator, so you may want to, in order to see it, you may want to slow it down. So the little wheel shown up here allows you to slow down the simulation so you can stop it during the progress of the simulation. So let's do that. I'll get rid of the graphs, so the rubbish bin symbol allows you to delete the graphs. The circle, green semicircle arrow is resetting the model parameters. So if we run that again, I'll slow it down a bit further. You can pause at any point. So the same icon that allowed you to start the simulation, if you click on that one again, it'll pause. If you click it again, it'll carry on. But you can go and change parameters if you want. So the parameter list down here reflects the current values of the parameters that are being solved. So you can see we're up to time 57.8. The value of x is 1.33. So I could go and change mu at that point if I wanted. I could go and edit mu and then carry on with the simulation. So what this is showing is the value of x versus t at the top, so it's an oscillator, and the value of y, which is the derivative of x, on the next panel, and then it's showing a state space diagram at the bottom. So I'll do that once more. And you can slow it down. It's a log scale for slowing it down at the top. And if you want to stop the simulation, the second icon is the stop one. So when you then hit run simulation, it starts from the beginning again. Whereas if you're pausing, you just click the same one to pause. Once you've run that simulation, if you want to export the results, if you click on this little icon here, which is CSV, uses the standard format. CSV is the output format. So if you click on that, it'll allow you. It'll bring up a dialog which allows you to then save that as a CSV file, Van der Pol data. And then you can use a spreadsheet to have a look at the individual solution. So that'll contain the complete solution for all variables. Okay, so I'll pause there to see if there's any questions from people before we go on to the next example.
So apparently there are no questions. I might have one. So I'm using a version for Mac OS X, and I have some difficulties to set up the scales of my graphs, so I cannot see the results. So I don't know if anyone, somebody has same problem. Otherwise, you can just move on maybe.
By the way, you can move graphs around with, as I'm doing now, with the mouse. You can also zoom in or zoom out, and you can reset to where you were. And if you hold the shift key down, you can actually find the location of a particular point. All of this is covered in the tutorial, so I won't go over all the features for manipulating the graphs when you solve for them. Okay, so I'll go to the... Okay, Jerome, shall I go on?
Yeah, please go on.
So let's go to the next example. One thing, I'm just going back now to the PDF document. I've listed in the document the various functions that you can use to represent various mathematical operators. We use square, x squared with SQR here, but there's a whole list of other ones there. Maybe one other thing I'll just mention that's shown in Fig 3. If we go back to the editing environment in the code, this is the CellML text view, which allows us to quickly enter those equations and check that they're correct. But the raw, if you click on the button, that shows you the encoding, the actual CellML encoding of that model in XML. So CellML is an XML, extensible markup language standard, as an exchange standard, and it uses MathML for the mathematics, and it separately deals with metadata through RDF, which we'll come to later, or it's in the tutorial. But you don't normally need to read the XML file, but there it is in case you want to look at it. And then one which is just uninterpreted by the XML. And then there's another button which we'll come to later in the tutorial, which is to do with annotating the terms, because generally it's good practice to try and create modular models that are well-defined modules that you can then import into building more complex models. And part of the importing process is facilitated if the components of a model are annotated against biological ontologies and biophysical ontologies. That's covered in the tutorial, and we won't get to it today, but just to let you know, that's where the button is that allows you to create annotations of a model. Okay, so I'm going to go back to the simulation environment, delete that model, so we start from scratch again, and then we'll go on to, I think we've covered all the panel buttons. I've been using those, so I think that's covered already. And I've covered the fact that the parameters are indicated, various types of parameter or variable are indicated by the colors of those little icons. So let's go to the next one, which is on page 8, section 4. And this is opening a CellML file from a local directory or a Physiome Model Repository. So if we go back to the OpenCOR environment, use your control spacebar to access the left-hand side. By the way, there's also help facilities shown on the right-hand side. So down the bottom, there's a file organizer. So I'll get rid of models in there at the moment. So your one will initially be blank, but when you've created a model, such as your Van der Pol model, you can drag and drop that one down to your file organizer, or you can drag one from the model repository. So what I want to show you now is really how to use the Physiome Model Repository to access models. So if you, from the start, you should see a list of, it's saying 566 exposures were found. So that's directly, if you're on the web, that will be directly accessing the Physiome Model Repository. The first button here is a button for cloning a workspace, and I'm not going to do that now. That's basically, if you find a model that's very similar to the one you want to create, you can clone it and use that as your starting point for your own model. But what I want to do now is just use this next button, which is a right-hand, the right hand of those two icons. If you click on that, what it does is it finds, and I'm just using the very first one on the list, it will find if there's a CellML model corresponding to that example, and then when you click on that CellML model, it will then load it into OpenCOR. Having loaded it in, you can use your control spacebar to give more real estate for that model, and then you can see all the parameters of that model from the Physiome Model Repository shown here. I'm not going to run this model because it would take a little while to set it up to talk about how to interpret it, but I just want people to be aware that they can access all of the 600 or so models in the PMR Physiome Model Repository can be accessed through OpenCOR by just clicking on the model. So I'll get rid of that and go back. And it's, if you want to just go to the website, if you go to Google, type in CellML, you'll see the CellML project. That shows you the website for the CellML project. And if you go to models, there's other tutorial help, by the way, on that website. But if you go to the model repository, this lists all the models on the repository, covering a very wide range of types of biology. So, for example, if you go to electrophysiology, there's well over 100 models there of various electrophysiological models that people have developed. And generally these models will have a diagram with them showing the components of the model. What we're working on, but is not in OpenCOR yet, is the ability to create these diagrams automatically from the CellML. So most of the ones that are on the website have been done by hand, but what we want to do obviously is to access automatic from the CellML. But I think that the most interesting thing for people to be aware of is that if you look at the views available to a model, you can click on one that says mathematics, and that will then take you to a page where it renders all the mathematics for that particular model. And this is quite a complex cardiac electrophysiological model. You can also go to code. So if you click on generated code, you can then go to various types of code that will, again, are automatically generated from the CellML. So this is showing you the Python code for that particular model. There's also MATLAB, Fortran, C and C++. So those are the views that are available for this particular model, and there's quite a lot of other functionality for the website that tutorial covers part of. It doesn't by any means cover all of the functionality, but we will increasingly add all of the functionality to the tutorial to let people know how to use the facilities on the website. In particular, the fact that there's workspaces, so you can collaboratively develop models under password protection until a model, for example, is published, and then you might want to make it available as an exposure, public exposure. So there's a lot of facilities on the website for collaboration. David Nickerson, who's very much driving the CellML Physiome project efforts in Auckland, is the person who, if people have questions about the actual features of some of these facilities on the workspaces on the site, he's the person to contact. Tommy Yu is the webmaster for the Physiome Model Repository. And any questions webself, go to webmaster, you'll see Tommy Yu. Okay, so I think that's all I want to say about the CellML repository, and the fact that you can access it directly from within OpenCOR. So if we go back to the tutorial, again, I'm just keeping an eye out in case anyone has any questions, but if not, we'll push on. So what I've talked about here in terms of clicking on these buttons is highlighted, is discussed in the tutorial. I'm not going to talk about the first order ODE model. I think that was mainly because I wanted to make sure that this tutorial was accessible for people who didn't have any background in modeling or mathematics really, or only high school level background. So there's just a discussion about first order equations and exponential decay on there. And there's a picture of a New Zealand $100 note which has a picture of a decay curve from a first order equation, because the Nobel laureate Ernest Rutherford is a New Zealander he was the guy that split the atom, but he was also the person that first discovered the radioactive decay time constant. And so the banknote shows that sort of picture of that decaying radioactive decay process. So the next model I want to move to is this one, the Lorentz. So this is now a third order system, 3 first order equations shown here, and it just has more interesting behavior, so it's worth having a little bit of a play with this model. So what you should do now is go back to OpenCOR and copy and paste the text from here or type it in. So if I do that I've already got it, I'll just drag it over. And if you look under editing text view, that's what you should see. So now we've got a model called Lorentz. The main component has t, x, y, and z variables. They're all dimensionless still, and they've got initial values of 0, 1, 1, and 1. And then there's 3 parameters, O and beta, which have initial values 10, 28, and 2 2/3. Then there's 3 equations. The first one, dx dt, is sigma times y minus x. The second one, dy dt, shown there, and the third one. So I'll just pause to let people get those equations into OpenCOR. Oh, we do have one question here.
Yes, we do have a question. So maybe I can just, this is Joel Castro, maybe I can just unmute his microphone. Joel, please formulate your question. Okay, so I will formulate it because I don't know if you cannot speak. So at the left side of my screen, I just see Cell Model Repository and not Physiome Model Repository, and I can't import directly the model in the 7 version, or I can change that.
Sorry, I don't know the answer for that. Maybe David Nickerson and Andreas is online, he might be able to.
Okay.
There we go. There's an answer from David. I'm getting.
That's okay. That's okay. It's solved. Someone okay. From the David Nickerson, so gave a clue.
Okay, so Joel's just responded. That's fine. That's fine. All right. So if we go back to the example with those 3, remember the test is that when you click on an equation, you see the mathematics for that equation. If there's anything wrong, if I take away something, you'll get an exclamation mark. And again, when you save it to your directory, you can only save it if you've got the syntax correct. There's no particular significance to the indenting, but it just helps to have it indented to show the structure of the model. And remember, you can add / with a comment. If you want to. Then if you go to back to simulation, in order to compare between models, some of the variables that you set up for displaying will still be there. To make things less confusing, I'm just going to remove those. You can untick them as well, but I'm just removing them. So we're starting from scratch, effectively. So with this model, what we're aiming to do is to produce 3 graphs, one which has X versus time, one which has Y versus X, state space description, and one that has Z versus X, another part of the state space. So as you can see from the figure 10, the start and end points that are convenient for this model are to run it for 50 time units and to set the increment as 0.001. So we'll change that to 50 and point interval to 0.001. Then for the first one graph, we go down to X, right click, plot against variable of integration. For the second one, right click on Y and plot against X. And for the third one, go to Z, right click against main, plot against X, I think it was. Yep. So we should have X as a function of time, the variable of integration, Y versus X and Z versus X. And you can check that if you have your graph panel open. So the top one is X versus T, next one's Y versus X, and the next one's Z versus X. Okay, so this time what I suggest you do is roll the little wheel well to slow it down very substantially so that you can watch the solution evolve. And then click Go. And now you can see that the graphs are rescaling to accommodate the current solution. That'll settle down in a minute. Perhaps go a little bit faster. And you can see that this is a chaotic system, so it's going to spin around more than one focal point. So perhaps I'll speed it up a bit more. So it's going around this critical point here. And it flips back to, and occasionally zips back again. And then if you, I'll just run that again, reset the parameters. It's just an interesting behavior, typical of a system that's very sensitive to initial conditions and to parameters. So if you change some of the parameters here, you can get quite different behavior. By the way, one extra panel in OpenCOR down the bottom, it shows you, gives you information about the simulation time. Most of that time will be plotting, the fact that it's continuously plotting to the screen as it runs. So, but that was with that slowed down. If we roll the wheel right over and run it, then that was taking 14 milliseconds to run. We've got some more questions here, Jerome. Should we deal with those? Oh no, sorry, those are the ones from before. Right. Okay, so I'm going to go back to the tutorial. And what I think I'll do now, we've only got 10 minutes left, and what I'm going to do is just innate some of the other what this tutorial is covering and where we're intending to go with OpenCOR, and also what we're currently adding to the tutorial as well, because we'll make it available as we improve it, or as we add. So the next sections in the tutorial deal with units. So CellML uses physical units. Most bioengineers and physiologists like to think about the quantities they're dealing with. Mathematicians tend to like to deal with dimensionless equations, but it's often convenient when you're thinking about the physics of a problem to be thinking about the magnitude of the terms and the units, the values of the units that you're using. CellML will automatically check that your units are consistent. So you know that when you're running a model, it's been checked for consistency of units. So the context that the tutorial is using to explain units is electrophysiology, through initially the background to the Nernst equation and the development of equilibrium potentials in giant squid axons and then in cardiac cells, and giving a little bit of explanation from the second law of thermodynamics about the derivation of the Nernst equation. And then it talks about the gating behavior of channels, so the idea that you're moving between the equilibrium potential for potassium in the rested state and then moving towards the equilibrium potential for sodium when the cell is activated, through a gated process that has time dynamics. And so what the next few sections do is to illustrate this for the potassium channel from the squid axon that Hodgkin and Huxley used, then the sodium channel, and then puts it together into the Hodgkin-Huxley equations. And then it's dealing with units, and SI units, there's only 7 of them: length, time, amount of substance, temperature, mass, current, and luminous intensity. All other units are derived from those 7. So CellML incorporates those units intrinsically and then incorporates a whole range of other standard units that build on those, such as Newton, Joule, Pascal, Watt, etc. And then it uses a standard terminology for indicating a multiple or fraction of a unit, and there's a list there shown for those. And then this example, or this box, shows you how to set up units based on those starting points. So for example, when you define a unit of milliseconds, so I'll make my screen a bit bigger for that. When you define milliseconds, what you're doing is defining a unit of seconds with a prefix of milli. Then when you define a unit which is per millisecond, i.e. millisecond to the -1, then you define seconds with a prefix of milli and an exponent of -1, and so on. So that sets up all the units for these ion channel models. And then the tutorial covers the development of the potassium channel models, and I'll just scroll through this, and then runs it as a single isolated module for the potassium channel, and then for the sodium channel. And then for the nerve action potential, it imports those channels to create a composite model. Yes?
Peter, just before we go through the nerve action model, so we had a hand raised by Muhammad Wazir Khalid, so maybe I will unmute him.
Okay.
Yeah, Muhammad, please, if you can talk.
Yes, can you hear me?
Yeah.
Yes.
Yes, it is, I don't know where to write it, but I was wondering, where can I get this tutorial? Is it uploaded on the website, or where is it?
It's Can you answer that one, Jerome? It's on the VPH website. It's on the CellML website.
Yeah. So you should have the link within the email that you've received from Martina Conti from the VPH manager. Otherwise, just type a question and then I can answer and I copy the link to you.
Okay, and I have another question as well. Hello?
Yeah.
I'm trying to model calcium ion concentration in astrocytes, brain cells. Do you think is it possible? Currently, I'm using console multiphysics. Do you think is it possible to model that in this software as well?
I didn't quite catch all the question. You're modeling an astrocyte.
Yes, calcium ion concentration in astrocytes. So, do you think is it possible to use model that kind of stuff in this software?
Yes, it is. If it's a system of ordinary and algebraic.
I'm using partial differential equations.
Yes, no, you can't. If it's partial differential equations, that means it's got a spatial context. Then it needs to use what we call FieldML standards. So no, it won't. The CellML is only for the ODE algebraic.
Okay, so this is limited to ODE only.
Yes.
Okay, thank you.
We will be extending this tutorial to cover other FieldML standard and the use of other software for partial differential equations, but at the moment we're just focusing on the ODEs.
Okay, thank you very much.
Okay. And I just want to put in a slight warning here for people that are trying the tutorial with the Hodgkin-Huxley equations. There's actually a mistake in the, I think I need to double check some of the code that's listed for the Hodgkin-Huxley, because we tried it, we double checked it earlier today and we found a bug, so just be aware of that. Sorry about that. The next one is a cardiac action potential model. This one does work, by the way, directly from the tutorial. And the main difference here is the fact that cardiac channels have a lot more control over the potassium to provide a plateau during which the heart contracts. But there's an example, and what we're particularly trying to emphasize in this example is the fact that CellML allows you to put all the units into a separate file and then import that file, and also put all your parameters into a file which you can import, so that you can have multiple files corresponding to different parameter sets that you might be needing for a particular model. But it also it's the fact it's very important to develop these models with a modular structure. So this is illustrating the fact that this Nobel 62 cardiac model is built on it first of all defines 3 submodules for the sodium channel, the potassium channel, and the leakage channel, and then imports those, encapsulates them as the example shows, and in order to then solve the equations at the membrane level. So that example is particularly important in terms of showing how to build, how to make best use of the modular structure, the import structure of CellML. And then the next part of the tutorial talks about annotation, and this is where you want to annotate the components of your model against ontologies. We will provide this, at the moment, the capacity within OpenCOR allow annotate against the biological ontologies, but we don't yet have the ontology for the biophysics, which is needed to complete this section. So that'll be a future development. And then, so there's an explanation of why ontologies are important and how it all works there. Then there's more information about the Physiome Model Repository, listing of all the models, an example of a particular model, more complex model. And also here there's an indication of where we're heading with this, which is to also take some of these models apart and allow you to link individual components, such as a particular protein associated with a transporter, in this case a sodium chloride co-transporter, where we can then link to bioinformatic databases, particularly UniProt and FMA databases for information about a particular protein. And expect to see more and more of this on the CellML site, where we're creating libraries of proteins as ion channels or transporters or enzymes that can be accessed as standalone modules for importing into other more complex models. There's a speed comparison there against MATLAB, and then there's a section on the use of a markup language for specifying the actual simulation. So the latest version, not the stable one that you're using, but the very latest version of OpenCOR is building in access to CellML, which is a simulation definition language, and the idea of providing functional curation or comparing models with each other and against experimental data. So there's an initiative from Oxford that is developing a capacity for that under a website called WebLab. So links to that and a description of that in that section. And then a little bit about future developments, which is the biosignal ML, the ability to handle signals in a standardized way with a standard for signals that has now been implemented in the beta release of OpenCOR, so that will appear fairly soon in the new release. And there's various other features that are being worked on by Alan Garny that are mentioned there. And we're hoping to have a lot more facilities around parameter estimation and system identification available as well. And the next part of the tutorial that I'll be building will deal with signal transduction pathways, as well as some whole body models of acid-base physiology. So I think that's it. I think that's all I wanted to talk about. What I hope you've got out of this is the confidence that you can core or know the basics of how to use it, and that you can access the Physiome Model Repository. And anyone that wants to keep up to date on new developments for both OpenCOR and the Physiome Model Repository should just let Martina know so she can add you to a mailing list so we can keep people posted. So unless there's any questions, I think that's probably all from me. Jerome, is there anything else you think we should say?
Okay, so thank you very much, Peter. So guys, if there is one question, then this is the opportunity. So you can use a hand up tool. So it seems that there are no specific questions from the attendees. I do have one, if I may.
Sure.
And so I'm not user of OpenCOR or CellML models. However, we do use agent-based simulations, and we're using, for example, softwares like Brevi or NetsLogo. And in the agent-based code, so you can program relatively simple ODs. So in order to establish your rules for a specific agent. And now what I'm seeing from your tutorial is that CellML would be extremely useful when the rule you want to code becomes to be mathematically very complex. So I'm wondering whether there is a way to efficiently, iteratively couple agent-based simulations and CellML models in order to
Yes.
to exploit the CellML mathematics.
Yes, no, I understand, and that is something that we've been thinking about, because there's no reason that agent-based models shouldn't fit into the CellML framework. The main the description of the rules or the statements that you make as part of an agent-based model, that's fine. The issue is more around the metadata, just how we handle the metadata for agent-based models. But we're very keen to do that because a lot of people use agent-based models, particularly as a way of moving between ODEs at the subcellular signaling level and agent-based models to represent the integrated behavior of a whole cell, for example, and then often plugged in into PDEs at the top end. So we are trying to facilitate that also. But at the moment, and I think what we'll do is, as we figure out how to deal with the metadata side of agent-based models, we'll create examples on the Physiome Model Repository to show people how that can be, how CellML can be used for those types of models.
Okay, thank you very much for the answer. So if there are no questions from the audience, and it seems there are not, so I think that we can close the webinar. Before we close the webinar, so I would encourage the attendees to write Martina emails if you have specific suggestions of speakers you would like to give a webinar for the next editions. And so I'm checking whether there are questions. No. So we are closing the webinar. So Peter, thank you very much again for your time. And I know you're on holiday, so so particular thanks. Okay, so thank you everyone to attend, and see you at the next webinar, hopefully.
Okay.